There are three major pathways that activate the complement cascade. The three of these major pathways that activate the complement cascade are the classical pathway, the lectin pathway, and the alternative pathway. All three of these pathways result in the C5 convertase, which then goes on to create the MAC. The classical pathway is the only one of these three pathways that is considered to be part of the adaptive immune system, which is due to the requirement of the antibodies in order to induce this pathway. Usually only complexes formed by the IgM or some subclasses of the IgG can activate this classical pathway, and components involved in this classical pathway are the C1, C2, C3, and C4 molecules. So the C1 molecule contains one molecule of the C1q and two molecules each of the C1r and C1s, as you can see on the picture to the left. The C1q molecule itself has 18 polypeptide chains and can associate to form 6 collagen-like triple helical arms, as you can see on these pictures. Each arm has a globular head at the end of it, and these globular heads are what bind to the Fc sites. The C1q needs to bind to at least 2 of these Fc sites in order to have a stable C1 antibody interaction. So it helps for you to remember throughout this process that the binding of one component to the next usually always does induce either a conformational change or some kind of cleavage that allows for the next reaction in the cascade to occur. So the binding of the C1q molecule to the Fc region induces the conformational change in one of those C1r molecules, which then cleaves and activates the other C1r molecule, and both of those C1r molecules cleave and activate the C1s molecule. Oops, sorry. Once the C1s molecule is activated, the fragmentation of the C4 can take place. So the C4 proteins are fragmented and produce the C4a and C4b. C4b must quickly bond to that target membrane surface. As you can see, it bonds within the vicinity of the C1 complex. If it does not bind quickly, then it's hydrolyzed and it can no longer bond to the cell surface. And about 90% of the C4b that is created is hydrolyzed and does not bond to the cell surface. C4b then binds to C2, which is then cleaved by the C1 enzyme to form C2b and C2a. C2a remains attached to the, sometimes will recombine with the C4b, and that creates the C4b2a, which is also known as the C3 convertase. The C3 convertase can hydrolyze the C3 molecules, which creates fragments, but it's not extremely stable because the molecules break down quickly, so it has to hydrolyze a lot of the C3 molecules quickly until it's stabilized by the C3b molecules. So this creates the C4b2a3b once the C3b molecule is attached to that. This is known as the C5 convertase. The C3 component of the C5 convertase binds C5 and allows for the C5 to be cleaved, which then allows for the C5 through the C9 process to be initiated, and that ends in the formation of the membrane attack complex, which is done at the end of all three of these pathways. The lectin pathway uses lectins as its specific receptors molecules. Lectins are proteins that recognize specific carbohydrate compounds, and they're primarily found on the microbial surfaces. It is the lectin pathway is part of the innate immune system, which is unlike the classical pathway, and it does not need antibodies to undergo this pathway. But it does also begin with the C1, C2, C3, and C4 molecules. Mannose binding lectin is one of the lectin receptors that can induce this pathway. It's considered to be the PRR, and people with low BLs usually suffer from repeated serious bacterial infections. So the MBL binds to sugars on the outside of the cells, which activates the MASP family. MASP2 is activated like the C1s to cleave the C4 and C2 molecules, and then it continues like in the classical pathway throughout the rest of that process. So basically the soluble lectin receptor takes place of the antibody as the antigen recognizing component, and the MASP takes place of the C1R and the C1s. The alternative pathway is also part of the innate immune system, and it does not rely on antibodies. But the early components of this differ from the lectin pathway and the classical pathway. These early components include the C3, factor B, factor D, and properdin. So the complement proteins in this pathway bind directly to the bacterial cell surface, and this usually does better in the gram-positive and gram-negative bacteria. The C3 component hydrolyzes spontaneously, and this happens naturally, to form the C3b and C3a. If the C3b lands on the surface of the bacterial cell, it becomes more stabilized, otherwise it hydrolyzes and goes off. The negative charge of the cell surface stabilizes that C3b component, and then factor B, which only binds to the C3b if it's on the cell surface, then binds, and it becomes the C3b big B. Then the factor D comes and cleaves off part of the factor B, leaving behind the C3b big B B. And the properdin that's on the cell surface helps to stabilize that molecule, the C3 convertase, which is that version of the C3 convertase is only seen in the alternative pathway, which this also acts like the C3 convertase that's seen in the other pathways. It doesn't act differently. So once the C3 convertase is stabilized with the properdin, it again begins to rapidly cleave C3 and generates large amounts of the C3b, much like classical and lectin. And these large amounts of C3b that have been cleaved bind with more factor B, which results in the alternative pathway C5 convertase, which, as you can see below, it is the C3b, little b, big B, little b, C3, little b. And this is also stabilized by the properdin. The rest of this pathway is then similar to the other two pathways, and it is carried out to form the membrane attack complex. So basically all three of these pathways, even though they have different things, come through and converge in the end to form the membrane attack complex, as you can see in both of these. And there you go.